Good evening. We'll be waiting for a couple of minutes for everybody to join, and then we will begin with our session. Good evening, everybody. Welcome to the ninth session of Genome Editing and Engineering. Uh, it is an NPTEL course taken by Professor Utpal Bora of IIT Guwahati. I am Sanket Mukherjee, a Prime Minister's Research Fellow at the School of Biological Sciences, IIT Delhi. So today's week's topic is genome engineered um, transgenics, basically disease models. So we'll come to the questions directly, and then we will see if there are any doubts. We'll clear them out as well. So coming to question number one, which of the following is not a characteristic of the animal model? Is it A, a close relative or closely associated with the host tissue distribution, disease progression, and similar route of infection, if not identical? B, the disease course should be relatively shorter in the animal model to complete the efficacy test reasonably and facilitating the rapid transition to human clinical testing. C, the animal models have to be non-reproducible. Or is it D, disease model animals should be easy to maintain, work with, readily available in adequate numbers, relatively inexpensive, and free of regulatory constraints? I'll give you a minute to think about it, and then we will discuss the answer. Okay, so the answer to this question would be C, the animal models have to be non-reproducible. It's that it's not like that. In fact, the animal models can reproduce, although in certain cases we do prefer male, mice or other kind of animals because of some of the hormones that are associated with pregnancy might affect, the testing. But in general, uh, animal model need not be non-reproducible. It needs to not be infertile. So that is the answer to this question. So what is a disease model? A disease model is an animal or a cell that displays all or some of the pathological processes that are observed in the actual human or animal disease. Studying disease model aids understanding of how the disease develops and testing potential therapeutic approaches. So what are animal disease models? All animal models, it could be mice, rats, zebrafish, and others, are non-human species, but they are sufficiently like humans in their anatomy, physiology, and also genetics. And the response to pathogen can extrapolate the result that we get in normal human physiology and disease. So they are used in medical research as they can mimic aspects of disease found in humans by using animal models. Researchers can perform experiments that would be impractical or ethically prohibited with humans. So numerous model organisms have been developed and are extensively used for studying basic biology and pathophysiology of human diseases and the development of novel therapeutics. So the we will dis I mean the um, lecture has already talked about the concepts of a good model design and its application, and the theory behind underlying biological modeling and the process of producing a valuable and relevant animal model. So these animal models are expected to, replicate the disease symptoms in humans. So development of model organisms have revolutionized our understanding of the mechanisms underlying normal development, adult homeostasis, and human disease. Many things we know about gene function in model organisms and its application to human has come from gene knockouts, knock-ins, mutation, single nucleotide polymorphism, and so on. So why do we need animal disease models? Because they, researchers, they can carry out experiments that would be impossible, impractical, and ethically prohibited in humans. For example, to test the effect of drug before using it on humans, to study the safety and efficacy of periodontal treatment modalities like regenerative procedures like bone grafts, surgical implants, for understanding the causes, the biology and prevention of diseases. That for that we require animal models. Numerous experimental and animal models. Sorry, numerous experimental animal models for diverse diseases have been successfully employed to screen new bioengineered chemical or herbal therapeutics that might have the potential for treatment of human patients. So there are more than 550,000 studies that have reported to use animal models for different diseases in NCBI alone. So what should be a characteristic of animal model? An ideal animal model should possess certain characteristics as a prerequisite. So let's see what those characteristics are. It should be a close relative or closely associated with the host tissue distribution, disease progression, and similar route of infection, if not identical. The closer it is, the better. The disease course should be relatively shorter in the animal model for completing the efficacy test in reasonable time and facilitating rapid transition to human clinical testing. There should be sufficient disease correlation and pathological equivalence in the model animal despite the genomic differences with humans. Disease model animal should be easy to maintain, work with, readily available in adequate numbers, relatively inexpensive, and free of regulatory constraints. The animal models have to be reproducible. So that is exactly what the opposite of this option was given. So the tests which are done in animal model could be reproduced further. That should be the aim. Now coming to question number 2, find out which of the following is not a widely used method for the generation of transgenic mice. Is it A, spontaneous mutations, B, retroviral infection, C, microinjection of DNA constructs, or D, sexually induced mutations? I'll give you a minute to think about it, and then we will be discussing the answer. Okay, so the answer to this question would be D, sexually induced mutations. This is usually not a method for generation of transgenic mice. We'll be discussing about the rest three. So why do we use mice as a model organism? Because one, the mice's genome is very similar to that of humans. A good genetic or molecular toolbox is available, and animals' small size facilitates large scale, high throughput studies, making it a cost effective model. Its potential for making medical research and in particular drug development more efficient could be increased by solving a range of identified bottlenecks. Mouse models have been successfully used to validate drug targets and to determine efficacious and safe dosage schemes for combination treatments in humans. These cases have one factor in common. They do not aim to fully model a disease or disease mechanism, but rather set out to obtain specific functional information. So, the most commonly used small mammal animal in cancer models are also mice, but there are several drawbacks. A typical human is 3,000 times larger than a mouse. It lives for 30 to 50 times longer, and therefore it undergoes about 105 more cell divisions in a lifetime. Now, mice develop cancers of mainly mesenchymal origin, such as sarcomas and lymphomas, even without genetic modification, whereas with age, humans have a bias towards development of epithelial cancers known as carcinomas. Due to the small size and short lifespan of mice, loss of certain tumor suppressor genes is insufficient to result in development of cancer in a highly penetrant manner, particularly when such mutations are heterozygous. Now let's talk about the widely used methods for generation of transgenic mice. So one of them is spontaneous mutations. These modifications appear spontaneously in mice colonies after successive breeding events and are usually detected when associated with a phenotypic change. The analysis of genetic background of spontaneously mutated mice can be associated with events encountered in human pathologies and further used as models for specific diseases. So, very like in case of humans, we have spontaneous mutational events. Similarly, mice also, there could be spontaneous mutation events that could also be induced by using certain mutagens, and that would essentially give you a pathological phenotype. That's the idea, and those are disease mice essentially. So we have a lot of mice models which are generated using spontaneous mutation. Then we have the chemical induced mutation. So these genetic modifications are based on the exposure of mice to mutagens by like ethyl nitrosuria, that can be used for large scale programs of mutagenesis and establishment of genetic alteration patterns responsible for human diseases. Widely used method for generation of transgenic mice. So here we give some sort of mutagen, which could be a carcinogen. It could be something that induces mutation in DNA, and that very specific mutation would lead to a diseased phenotype. So that's the idea. Now, retroviral infection, this is very similar to using retroviruses for gene delivery, very similar to that. In this case, we want a disease to happen. So this is one of the first partially controlled protocols for generation of transgenic mice and is based on the transfection of pre-implantation embryos with a retrovirus that contains the gene that needs to be replaced or modified. The modified embryos are implanted into the recipient females and are analyzed for the presence or absence of genetic modifications in concordance with the developed phenotype. So here we use retroviruses. Um, After that, we have microinjections as well. It is essentially injecting the DNA constructs into the fertilized embryo. And it is followed by transparent recipient females and analysis of the presence or absence of the genetic modification in concordance with the developed phenotype. So these are the four options how we usually see how transgenic mice are developed. Now, coming to question number 3, Janish and Mins created the first ever mice with transgenes in the year 1981, 1974, 1994, or 1954. So I'll give you a minute to think about it and then we'll discuss the answer. Okay, so the answer would be that Janish and Mintz, they created the first ever mice with transgenes in 1974. So if you look at the lecture slides, there was this was mentioned that Janish and Mintz, they created the first ever mice with transgenes in 1974. Although the mice could not pass the transgene to its offspring, the impact and applicability of these experiments were huge in spite of its limitation. So the word transgene was first coined much later in 1981 by J.W. Gordon and F.H. Rudel, who successfully incorporated the foreign gene in the host chromosomal DNA and demonstrated the Mendelian distribution up to two succeeding generations of progeny. Similarly, after that, in 2013, the first gene knockout mouse using CRISPR-Cas9 was created by Rudolf Janisch team, the same team, and Wang and co-workers. They injected TET1, TET2 single guide RNA with Cas9 mRNA into the zygotes, resulting in production of mice that carried 80% mutations in both genes. This was the first ever demonstration of using CRISPR-Cas9 system for one-step generation of animals carrying mutations in multiple genes. Now, coming to question number 4, select the correct option about humanized animals, which are 1, the mouse with its short generation time, 2, animal that developed from human, 3, they are especially transgenic animal models that mimic human responses to study. So is it option A, 1 and 3, option B, 2 and 3, option C, 1 and 2, or D, none of the above? I'll give you a minute to think on this and then we will be answering. We'll discuss the answer. Okay, so let's see what are the correct options. The mouse with its short generation time, that is essentially that should be one of the criteria of humanized animals. And animal that develop from human, that is not necessary that such criterion must be there. But yes, they are especially transgenic animal models that mimic human responses to study. That is the idea behind humanized animal models. So 1 and 3 are the correct options. So option A is right. Let's see what do we have to see. Discuss about humanized animals. So, labs, they resorted to developing simpler and effective models, especially transgenics, that mimic human response to study and understand various aspects of infectious agents, pathogenesis, disease progression, nature of protective immunity, and vaccine development. So in with respect to mouse genome, what were the modifications that were made? So the mouse with its short generation time and comparatively low maintenance and production cost is the perfect mammal for probing the genome to understand its functions and complexities. For improved understanding of human health and disease, researchers create a wide variety of mouse models that carry human DNA. The advances in genome engineering make possible targeted replacement of mouse genomic sequences with human orthologous DNA. So we call these as homologs. There are certain genes which have homologs in the mice. So we will basically replace the genes in the mice with the ones with their counterparts in humans. The capability range from finely tuned humanization of individual nucleotides and amino acids to incorporation of many bigger bases of human DNA. Let us see some emerging technologies for targeted genome humanization of mice. We will discuss the spectrum of genomically humanized mouse models and insights that such models have provided. So within the humanization procedures, well, there could be sublethal dose irradiation and humans blood cell injection, CD34 plus, I mean, positive human hematopoietic stem cells with bone marrow umbilical cord. These were, injected, or cord blood mononuclear cells, or there could be a human blood which could be directly injected. That could also be there. Um, Use of genetic engineering to create knock-in human genes. So basically, we want human genes to be functional in mice. That is also preferred. Like in case of interleukins, cerebrospinal fluid, thrombopoietins, and others. So then the next step involves human tumor tissue engraftment into using microsurgery techniques. Normally, the humanization process takes around 4 to 8 weeks until validation. Humanized clinical models are explored to get predictive results regarding the cancer therapy and can also be integrated in co-clinical trials. Now, compared to the large number of classical transgenic mice strains that contain mostly human DNA as randomly inserted multi-copy transgenes, the number of targeted genomically humanized mouse strains is less. So the latter are designed to have greater physiological relevance than their classical transgenic counterparts, as they maintain the correct genomic context of a gene of interest to preserve physiological expression levels. They have a correct spatiotemporal expression patterns. What does that mean? That the expression of these genes are regulated through tissue specific expression. So let's say there are some genes that are only expressed in hepatocytes and not in the neuronal cells. Such kind of expressions are conserved in mice as well. The translated protein is expected to display the unique biochemical properties of human gene, including potentially unique deleterious properties when mutated. When non-coding human sequences are incorporated, human-specific regulation, human gene splicing isoforms may also be maintained. That's the idea. So let's come to question 5. Find out the non-stem cell type from the following. Is it A, unipotent, B, pluripotent, C, totipotent, or D, multipotent? I'll give you a minute to think about it and then we'll discuss the answer. Okay, so the answer to this question would be the non-stem cell type is multipotent. So there is no such word as multipotent. In fact, stem cells could be unipotent. That means they can only differentiate into one kind of lineage. They could be pluripotent. That is, they could form all the three germ layers apart from, I mean, they just cannot form the placenta, and a totipotent stem cell can also form a placenta along with the three germ layers. So let's talk about it. So cell, tissue, organ, and body, all our body cells, they arise from a single source. So there's a zygote, which then, it further divides into various cells, and it forms the entire body in the entire human organism. Our body have 200 different cell types. Each type of cell is specialized to carry out a particular function, either alone. For example, RBCs, they function alone, or usually by forming a tissue. Now, the different tissues then combine to form specific organ, which are like factories where every type of cell has its own job. Same genes, yet different phenotype and functions are, performed by different kind of cells. So essentially, the gene that you will have, or the chromosomes, the DNA that you will have in a liver cell will be exactly the same as what you will have in a nerve cell, or what we would be having in any other, maybe the muscle cells. But their function is different, their phenotypes are different, they are even morphologically different. So this kind of regulation is usually achieved through epigenetic. That is what is widely believed. But anyway, um, so all these 200 different cell types within an individual human organisms are genetically identical. The variation in the cell types arise due to differential regulation of the expression of genes. Now, uh, if we talk about the basics, so stem cells, what are these? Stem cells are unspecialized cells, or we could rather say undifferentiated cells, which are able to change and transform into other types of cell form found in the body. This process is called cell differentiation, and it leads to the development of all types of cells in the body. In nature, stem cells are produced only from stem cells. So that's how it happens. So stem cells are able to differentiate into any cell of an organism and have the ability to replicate, uh, and self-replicate, and they can renew themselves. Basically, stem cells are of the following types. They could be unipotent, oligopotent, multipotent, pluripotent, and totipotent. And if you see the plasticity of these stem cells, so this would be the order. So the totipotent can essentially form totipotent cells. They can form an entire organism, including the placenta. Pluripotent cells, they can form all the three germ layers, the ectoderm, mesoderm, and endoderm, but they cannot form the placenta. Multipotent stem cells, they can differentiate into various lineages. Oligopotent is also very similar to multipotent, but again, the lineages are small in number. Unipotent stem cells, they can only differentiate into a particular cell type. That's the idea. So there are several steps of specialization, or we could rather say differentiation, and the developmental potency is reduced with each step. For example, a unipotent stem cell is not able to differentiate into as many cell types as a pluripotent one. So unipotent stem cells are characterized by the narrowest differentiation capability and a special property of dividing repeatedly. The latter feature makes them a promising candidate for therapeutic use in regenerative medicine. These cells are not only able to form one cell type. These cells are only able to form one cell type, let's say like dermatocytes or hepatocytes or lymphocytes. So there's only one kind of cell type that they can form. Only oligopotent stem cell can differentiate into several cell types. A myeloid stem cell is an example that can divide into white blood cells and red blood cells, and not red blood cells. Now, multipotent stem cells, they have a narrower spectrum of differentiation than the pluripotent stem cells, but they can specialize in discrete number of specific cell lineages. One example is hematopoietic stem cells, which can develop into several types of blood cells. After differentiation, a hematopoietic stem cell becomes a oligopotent cell. Its differentiation abilities are then restricted to cells of its lineage. However, some multipotent cells are capable of conversion into unrelated cell types, which suggest naming them pluripotent cell. So yeah, after that we have the pluripotent stem cells, which they form all the germ layers, but not the extra embryonic structures, exactly such as the placenta. Embryonic stem cells are an example Embryonic stem cells are derived from the inner cell mass of the blastocyst, pre-implantation embryo, that is the blastocyst. Another example is induced pluripotent stem cells, which have a very promising potential because for creating induced pluripotent stem cells, what we can do is we take a somatic cell, a completely differentiated cell, let's say a skin cell. We take that cell and then we inject with some transcription factors. You must have studied about the Yamanaka factors. So if we give those factors to these differentiated cells, so they will go back to their previous state, and they will form induced pluripotent stem cells. So that has a very promising approach in regenerative medicine. Then there are totipotent stem cells. They are able to differentiate and divide into cells of the whole organism. That also includes the placenta. One example of totipotent cell is a zygote, which is formed after a sperm fertilizes an egg. These cells can later develop into any of the three germ layers and or form a placenta. After approximately 4 days, the blastocyst inner mass becomes pluripotent. This structure is the source of pluripotent cells. So let's come to the next question. The important considerations while using iPSCs as a disease model are: one, whether the disease is a monogenic or polygenic disease; two, the penetrance of the mutations; three, the age of onset; so, or four, whether differentiation into an appropriate cell type is possible. So will it be A, 1, 2, and 3, B, 2 and 3, C, 1 only, or D, all of the above? I'll give you a minute to think on this and then we'll discuss the answer. Okay, let us look at each of the considerations. So for using iPSCs as a disease model, so whether the disease is monogenic or polygenic, of course, that is one consideration that we are going to see. What does that mean? Whether the disease is caused by one gene or multiple genes, that's what we need to see. The penetrance of the mutation, why is that important? Because um, let's see it in a way where you have a disease which is because some of the cells have a mutated gene, let's say, but not all cells have the mutated gene. So that is what is meant by the penetrance of the mutation. The age of onset, yes, that is also very important because with age, the repairability of the cell, they drastically reduce, and the mutations, they tend to accumulate when you are older. So yes, age of onset is also important. Uh, Whether differentiation into appropriate cell type is possible, yes, of course, iPSCs are used so that they can differentiate into any cell type. So that is also important. So the answer to this question would be D, all of the above. Let's see what do we have. So for the considerations for iPSC disease models are that whether the disease is monogenic or polygenic, the penetrance of the mutation, the age of onset, whether differentiation into appropriate cell type is possible, or if there is an appropriate phenotypic readout at a molecular or cellular level. What does that mean? Well, see if there is a disease, there needs to be some molecular markers to that disease so that you can identify the diseased phenotype. So uh, if when you have given a proper, when you're treating the disease using iPSC, so you need to see whether these markers are getting lowered or they are getting higher. So those, for that we need to have a proper phenotypic readout at the molecular or cellular level. So molecular markers could be one of them. Cellular markers could be the let's say some of the genes that they express, some of the proteins that they are expressing, those kind of things we can see. It is critical why with any iPSC disease model to pinpoint a cell type in which the disease manifests, to be able to differentiate effectively into those kind of cells and to identify a molecular or cellular phenotypic readout of the disease state. Differentiation protocol are now available to efficiently generate a large variety of lineages, and many others are being developed by using cocktails of small molecule inhibitors or transcription factor over expression. Now, such protocols often result in a mixed population and purification of the desired cells, for example, by fluorescence activated cell sorting using an appropriate marker or reported gene can be used to enrich the population of interest. Another important consideration with using iPSCs in the disease modeling is that these cells and the differentiated derivatives often resemble those of fetal origin. And therefore the age of onset of any disease becomes relevant because, yeah, these are the options basically. What are the limitations? Although the benefits of iPSC technology are undeniable, there are some limitations in their use for modeling certain disease states. Such in vitro models have immense power in terms of stability, scalability, and being able to apply techniques such as high throughput genetic or pharmacological screening that would not be possible or be technically difficult in an in vivo setting. However, they are limited in their ability to recapitulate complex tissue architectures, both in terms of complexity of cell types as well as their spatial organization, making analysis of many physiological or system level phenotypes challenging. So what does that mean? Is that every a single tissue, every single organ, they have a very rigorous, you know, arrangement, and replicating that arrangement even when the cells have differentiated is difficult. So highly defined co-culture systems can be beneficial in some situation, for instance, where the effects are non-cell autonomous or rely on cell-cell signaling. Most genetic diseases are due to small contribution from a large number of genes. Such polygenic disorders are inherently more difficult to study than monogenic disease, as the penetrance and severity of the phenotype due to any single mutations are lowered. Genetic targeting strategies in combination with efficient differentiation protocols will provide a powerful tool for autologous iPSC-based therapies in the future. One of the major limitations that we still deal with iPSCs is their low reprogramming efficiency. So when you give a cocktail of transcription factors in a hope that a skin cell will transform into an iPSC, the number of cells that actually do transform is very, very less. So that's a problem as well. So this is the summary. You take the patient, you take some specific somatic cell, and you then give the reprogramming factors. These specific factors. They are called the Yamanaka factors or the Thomson factors, depending upon who discovered it. So yeah, that's the thing. So when you give these factors, you expect that iPSCs are developed. Now these iPSCs which have, let's say, the faulty genes, you will correct the gene here using, let's say, CRISPR, or which is the most used method, or any other genome editing method, and then you will differentiate the cell into the cell type you want. So that's the idea behind this. Coming to question number 7, to induce T1122Q 24, 12 dash are expressed in human embryonic stem cells, sorry, HESMP cells to create double stranded break in both genes. Is it ZFNs, CRISPRs, TALENs, or meganucleases? I'll give you a minute to think about it, then we'll discuss the answer. Okay, so the answer to this question is zinc finger nucleases. They have been used, they are expressed in these cells to create double-stranded breaks. Let's see why are these studied? What are these? So, Ewing sarcoma family of tumors, ESFT, is the second most common type of primary malignant sarcoma. ESFT includes soft tissue Ewing sarcoma, peripheral primitive neuroectodermal tumors, and Askins tumors. Ewing sarcoma is a bone-associated malignancy arising primarily in childhood and adolescence. It is an aggressive cancer harboring a characteristic translocation. So here the translocation is in between the chromosome 11 and 12, where 11 and 22, where the Q24th part of the 11th chromosome and the Q12 part, they get translocated. So if you do not know what P and Q are, so in a chromosome, if these are the centromeres, then there is a short, usually a short, arm, and there is a long arm. So the short arm is the P, the long arm is the Q. So, and these locations like 24 or 12 are the locations basically in the chromosome. So this is a translocation event that happens. The same has been explained here. So over 90% of Ewing sarcoma contain a translocation which fuses the EWS gene on chromosome 22 with the FLI1 gene on chromosome 11. So the figure explains how it looks like, and then uh, Ewing sarcoma translocation involve breakpoints within the EWSR and FLI1 gene on chromosome 22 and 11, respectively, creating an EWSR1 FLI1 fusion gene on DR. So the EWSR1 FLI1 fusion gene product is a chimeric transcription factor that initiates an oncogenic transcription program, and that oncogenic transcription program leads to development of cancer. That's as simple as that. Now, so induction of a T1122 translocation in these cells using zinc finger nucleases was performed. So zinc finger nucleases, they were expressed in these cells to create double-stranded breaks. So the zinc finger nuclease, EWS, and zinc finger nuclease FL1 cleavage. So basically cleavage sites are within EWSR and FLI1 introns. So they do not affect the expression of these genes relevant to the EWSR1 FLI1 translocation. Zinc fingers in the ZFNs are designed to bind to the shaded sequences, as you can see in the arrow. The presumed the double-stranded breaks sites after the fork point nuclease domain cleavage ZFN cleavage activity in HES-MP cells are monitored by T7 endonuclease assay. The region around zinc finger nuclease site is amplified. The amplified products is then denatured, reannealed, and then subjected to T7 endonuclease cleavage. And insertion and deletion characteristic of imprecise DSB repair by non-homologous end joining give rise to T7 endonuclease cleavable DNA. Now, the nested PCR is done to detect the derivative chromosomes of DR11 and 22 in HES-MP cells, and translocation breakpoint junctions are only detected after expression of both the zinc finger nuclease EWS and zinc finger nuclease FLI genes. Then RT-PCR detection of EWSR and FL1 fusion transcripts after the zinc finger nuclease expression in HES-MP cells. The forward primer overlaps the exon 2 and 3 junction, and the reverse primer is at the exon 9 of FLI1, amplifying most of the entire both of the genes basically. That's what happens. Now let's come to question 8. Talents were designed using TALE, tail NT software, and the criteria used are the following except one. So is it A, talent binding sites range from 15 to 19 bases? B, the spacer length was 15 to 16 base pair to fit the Goldie talent designs. C, when possible, talent target sequences were selected around a restriction enzyme site. Or D, single clones were trypsinized and transferred to 24 well plates for the further selection by gene mutation analysis. So I'll give you a minute to think about it, and then we will be answering. I mean, we'll discuss the answer. Okay, so the answer to this question would be talents were designed using the tail NT software, and the criteria used are the following. So tail mining sites range from 15 to 9 base pairs. Yes, that was one criteria. The spacer length was 15 to 16 base pairs to fit the Goldie talent design. Yes, that was also a criteria. When possible, tail target sequences were selected around a restriction enzyme site. Yes, that was also a criteria. And D, single clones are trypsinized and transferred into 24 well plates for the further selection for gene mutation analysis. No, that was not a criteria. So the option would be D. So let's see the tail mediated somatic mutagenesis in murine models of cancer. So in this study, Zhang and associated used genome editing tools to create and analyze targeted somatic mutations in murine models of liver cancer. Talent were designed against beta-catenin gene and APC, two commonly mutated genes in hepatocellular carcinoma, to generate isogenic hepatocellular carcinomal cell lines. Both mutant cell lines exhibited evidence of WNT pathway dysregulation. Talents targeting beta-catenin promoted endogenous hepatocellular carcinoma, carrying the intended gain of function mutations. However, talents targeting APCs were not as efficient in inducing an in vivo homozygous loss of function mutation. They hypothesized that hepatocyte polyploidy might be protective against talent-induced loss of uh, heterozygosity. And indeed, APC gene was less efficient in tetraploid than in diploid hepatocytes. To increase efficiency, they administered adenoviral APC talents and could achieve a higher mutagenesis rate in vivo. Now, talents, they were designed using the tail NT software, and the criterias where the talent binding sites range from 15 to 19 base pairs. The spacer length was 15 to 16 base pair to fit the Goldie talent design. And when possible, talent target sequences were selected around a restriction enzyme site. So to construct talent plasmids, intermediate arrays were produced for each talent pair and that were compatible for golden gate cloning. Arrays were joined in the plasmid PC Goldie talent vector as below. So there was 150 nanogram each of PFUSA, PFUSB, PLRX, and 70 nanogram of PC Golden talent. So vector backbone and were mixed in a 20 microliter digestion ligation reaction, including 1 microliter of T4 DNA ligase and 1 microliter of ESP31. So these are the enzymes. Now the reaction was incubated in a thermocycler for 10 cycles of 5 minutes at 37 degrees Celsius, 10 minutes at 16 degrees Celsius, and 37 degree for 15 minutes, and 80 degrees Celsius for 5 minutes. 2 microliter of each reaction was transformed into E. coli and plated on LB ampicillin plates. Adenoviral APC talents were subcloned by cutting APC talents from the PC golden talent construct with SP1 and EcoR1 to transfer the full gene expression cassette to the adenoviral vector. The APC talent adenovirus was generated by molecular biology vector core UPSW. So then cell culture and transfection was done. So in H2.35 cells, they were transfected with 2 microgram of each talent arm in 6 well-plated well plates using lipofectamine. So lipofectamine is an agent that helps in transfection. And they were cultured for 48 to 72 hours before performing assays. So for selection of single APC mutated homozygous clones, H2.35 cells were co-transfected with APC talent pair and 0.5 microgram pcDNA 3.1 GFP, followed by cell sorting for GFP positive clones. Plating for extended culture and colony selection in 15 centimeter dishes. Single clones were trypsinized and transferred to 24 well plates for further selection by gene mutation analysis, and then xenograph experiments were performed. So now nude mice, they were injected with these clones, and then they were suspended in the 1:1 ratio of serum free media, and then the 5 tumors for each cell type were inoculated. Tumor volume was calculated according to the formula. And that's the experiment that is all about. So that's all that we have to discuss today. If you have any questions, you can definitely ask. Um, Otherwise, we'll just go through the questions once again, and then we can call it a session. So if there are any questions, you may raise your hands or you can speak if there are any questions regarding the content. Okay, if there are none, we will just go through the questions once again, and then we will end the session. So question number 1 was about which of the following is not a characteristic of the animal model, and the animal models have to be non-reproducible. That is obviously not a characteristic. Then about question number 2, which of the following is not a widely used method for generation of transgenic mice? Then sexually induced mutation is the answer. Question number 3, Janish and Mintz created the first ever mice with transgenes in 1974, but again, these transgenes were not um, transferred to the offsprings. Question number 4, which of the following select the correct option for humanized mice? So it is option A, 1 and 3. That is, a mouse needs to have a short generation time, and they are transgenic animal models that mimic human responses to study. Question number 5. The non-stem cell type from the following is multipotent. There is no such word as that. The rest are stem cell types. Question number 6, the important considerations while using iPSCs are as a disease model are all of the above, whether the disease is monogenic or polygenic, penetrance of the mutation, age of onset, and whether the differentiation into an appropriate cell type is possible. All of them will be the answer. Let's talk about question 7. To induce the translocation, dash are expressed in HES-MP cells to create double-stranded breaks in both genes. So that would be zinc finger nucleases. And then we have question 8. Talents were designed using the Talent NT software, and the criteria used were all the following, like Talent binding sites range from 15 to 19 base pairs, spatial length was 15 to 16 base pairs. When possible, talent target sequences are selected around a restriction enzyme site, but it was not that the single clones were trypsinized and transferred into a 24-well plate for the further selection by gene mutation analysis. So that's all about today's questions. If you have any questions, you can ask, or you may leave if there are no questions. Okay, thank you. I think that's all for today's session. We will be meeting next week at the same time for week 10. So all the best for your assignments and bye bye.